Imagine a patient, someone who successfully beat breast cancer. They went through the grueling treatments, the surgery.
All of that uncertainty and fear.
Yeah, exactly. Yep. All of it. And they finally get the all clear. They start the really hard work of piecing their life back together. But then years later, they get this just devastating scan. The cancer didn't just come back. It spread to the brain.
Right, which is what we call breast cancer brain metastasis, or BCBM.
And clinically speaking, it's a severe condition, right?
Fun.
I mean, we are talking about a median survival time of just 16 months. So welcome to the deep dive.
Thanks for having me.
Today, we're taking you inside what biologists used to call a biological fortress. We want to see how our bodies fight back against some of the most aggressive odds imaginable.
It really is a devastating diagnosis. And, for the longest time, the medical community looked at the brain and just thought of it as this impenetrable immune fortress.
Right, because of the blood-brain barrier.
Exactly. We believed the blood-brain barrier, which is essentially this tightly packed, specialized layer of endothelial cells and astrocytes protecting the brain, we thought it basically locked the immune system out.
So the assumption was, if a tumor managed to sneak past that barrier, it could just grow completely unchecked.
Yeah, unchecked by our body's natural defenses because the immune cells simply couldn't get in, or so we thought.
Right, like an impenetrable castle. But here's the thing for everyone listening. Is it really a fortress? Or is that just what we assumed because we literally didn't have the tools to look closely enough?
That's the exact question that sparked this whole shift in how we understand it.
Okay, so let's unpack this. Our mission today is to completely deconstruct this landmark 2026 study published in the journal Cancer Cell by Jasowitz and colleagues.
It's an incredible piece of work.
It really is. They analyzed this massive cohort of 156 human breast cancer brain metastasis tissues. And they didn't just want to see if immune cells were present, right?
No, they went much deeper. They wanted to uncover exactly which immune cells are fighting the tumor inside the brain, like the exact mechanisms of how they operate.
And how their specific locations translate to whether a patient actually survives.
What's fascinating here is just the sheer scale and the unprecedented precision of this study. But before we can identify the specific heroes of the immune system, we have to understand the battleground itself.
Right, the environment. Because BCBM is not rare, is it?
Unfortunately, no. It occurs in up to 32% of metastatic breast cancer cases. And these tumors, they are incredibly adaptable.
Adaptable how?
When about 15% of patients, the tumor actually changes its biological identity when it moves to the brain.
Wait, I need to pause on that. The cancer changes its identity. How does that even work?
It's driven by evolutionary pressure. So, for example, a patient might start with a luminal primary breast cancer.
Meaning that cancer cells have receptors for estrogen or progesterone.
Yeah, exactly. They are driven by those hormones.
Yeah.
And we have great drugs to block those hormones. But when the cancer spreads to the brain, it can mutate and just drop those receptors.
Wow.
Yeah, it switches to triple-negative breast cancer or TNBC. Suddenly, all those hormone therapies you were using, they're useless.
So the tumor essentially sheds its old uniform and puts on a new one that is much, much harder to target.
Exactly. It's mutating as it breaches the castle walls.
Then reading through the study, it seems like the location inside the brain matters immensely, too. It's not just about getting into the brain, but where in the brain.
Right. The researchers brought forward this brand new finding, tumors specifically located in the cerebellum. The structure at the very back of the brain responsible for coordination, those have a uniquely dismal prognosis.
Because for a long time, we thought of the brain as just one uniform environment, right?
We did. But the cerebellum has unique properties, most notably a really high level of GABAergic signaling.
Let me stop you right there. GABAergic, that's related to the neurotransmitter GABA, right?
Sure.
The chemical that usually calms the central nervous system down.
That's the one.
How does a calming chemical help a tumor? I mean, that sounds counterintuitive.
It's a brilliant connection to make. Yes, GABA is an inhibitory neurotransmitter. It slows down neuronal activity so you don't overstimulate. But in the context of a tumor microenvironment, that exact same calming signal can be exploited.
Exploited by the cancer cells.
Exactly. The cancer cells use it to suppress the local immune response. It effectively tells the incoming immune cells to just power down. It creates this uniquely hostile immunosuppressive niche.
Wow. They're literally using the brain's own relaxant to put the immune system to sleep.
Yeah, it's pretty insidious.
Okay, so how do we even begin to map a battleground that complex? I mean, to look at 156 patients and figure all this out, they couldn't have just used a standard microscope.
Oh, they couldn't. They used an arsenal of really futuristic tools. First off, they use highly advanced tissue psychometry.
Which does what exactly?
It allows them to count and identify cells in a tissue slice by using these fluorescent probes. The probes latch onto specific protein markers on the surface of the cells.
So it's like a really high-tech roll call. But then they took it a step further with single nuclei RNA sequencing, or snRNA-seq. Let me see if I can workshop an analogy for this, because this concept is wild.
Go for it.
Okay, so if I'm trying to figure out what's in a complex fruit smoothie, the old way, bulk RNA sequencing, is like tossing the smoothie into a chemical analyzer. It tells you there's strawberry and banana DNA in there, but it's just a mush.
Right, you lose the individual fruit.
So is the single nuclei sequencing like magically pulling the intact strawberry back out to examine it?
You're very close, but it's even more precise than that. It's like pulling out the strawberry, opening its cells, and reading the exact genetic recipe book that the strawberry was actively using at the moment it was frozen.
Wait, seriously?
Yeah. By isolating just the nucleus of individual cells, they can sequence the RNA to see exactly which genes are turned on or off in that specific cell, all without destroying the delicate surrounding tissue architecture.
And that architecture is everything, isn't it? Knowing the recipe is great, but in a war zone, location dictates strategy. Which brings us to the third tool they used, spatial transcriptomics, specifically molecular cartography.
Yes. Think of it as mapping the entire city down to the literal street address.
Okay.
They take a glass slide with a slice of the tumor, and they map those RNA transcripts to exact physical coordinates. It tells us not just who is at the party, but exactly where they are standing.
The ultimate Google Maps for cells, so you can see if the immune cells are deep inside the tumor islands or if they're just hovering on the edges, locked out of the fight.
Exactly, and using these tools, they definitively shatter the whole immune fortress myth.
Right, because T cells, the cytotoxic assassins of the immune system, they absolutely do cross the blood-brain barrier.
They absolutely do. But the density varies wildly from patient to patient. Some patient tumors had as few as 6 T cells per square millimeter, while others had up to 874.
That is a massive difference. Okay, so if we step back for a second and synthesize what we've discovered in this first layer for you listening, BCBM is highly variable. The tumor can literally change its subtype to evade drugs, and tumors in the cerebellum exploit calming neurotransmitters to become deadlier.
Right, and we now have the spatial and genetic tools to map every single cell.
Exactly. Which brings up a huge question for you listening. If T cells can successfully cross the blood-brain barrier and infiltrate these brain tumors, why don't they always win the fight?
And that is the exact question the researchers ask next, because just knowing T cells are present, well, it isn't enough.
Okay.
We need to know their exact job descriptions and the biological neighborhoods they live in.
So using all that spatial data, they clustered the 156 tumors into 5 distinct immune ecotypes, right?
Yes. You can think of an ecotype as a highly specific ecological system within the tumor.
Right. I noted that ecotype 1 was basically an immune desert. Almost no immune cells made it in, which is just a terrible prognosis.
Very poor outcomes for those patients, yeah.
But ecotypes 3 and 5, those were highly favorable for patient survival. Let's look at ecotype 3 first. This is where we meet the first major hero of this deep dive. CD8 plus TRM-like cells. Unpack that acronym for us.
Sure. TRM stands for Tissue Resident Memory Key Cells. And the CD8 plus just means they are the cytotoxic kind, the actual killer.
And these were a big deal in the study.
Huge. These cells were the defining feature in about 11% of the brain metastases studied, and they are the absolute key players of immune control. To understand why they are so lethal to the tumor, you have to look at the specific proteins they express.
Okay, tell me about the proteins.
Well, they have very high levels of activation markers and a crucial protein called CD103.
CD103. This is the mechanism that fascinated me the most. I like to think of CD103 as a molecular anchor.
That's a good way to picture it.
Yeah, because if standard T cells are like circulating police officers driving through a neighborhood on patrol, these TRM cells are completely different. Because of that CD103 anchor, they act like local bouncers.
I love that. Local bounce.
They literally glue themselves to the tissue, take up permanent residence, and memorize the specific local threat.
It's a perfect analogy. And because they use that molecular cartography, the Google Maps tech we talked about, we know exactly where these bouncers stand. They sit intratumorally.
Meaning inside.
Yes, they're anchored deep inside the actual tumor beds, face-to-face with the cancer cells.
But wait, I'm stuck on something. If they are right inside the tumor, surrounded by cancer cells, wouldn't the tumor try to shut them down? I read they engage in something called TGF-beta signaling. Doesn't the tumor usually use TGF-beta to suppress the immune system?
It does. Transforming growth factor beta, or TGF-beta, is normally a suppressive signal. Tumors pump it out to essentially tell the immune cells to stop proliferating and calm down.
So how do the TRMs survive that?
Here is the incredible evolutionary mechanism. These TRM cells have adapted. When the tumor hits them with TGF-beta, the TRM cells actually use that specific signal to upregulate their production of CD103, that molecular anchor.
That is incredible. The tumor is trying to shoot them with a tranquilizer dart, and the TRM cells use the dart to build a stronger residency permit so they can stay and fight.
Precisely. They turn the tumor's own weapon against it. It's brilliant biology.
But what happens to the T cells that don't have that CD103 anchor? Because not all of them do, right?
Right. And that's where the contrast becomes stark. The researchers identified another type of T cell called GZMK plus CD8 plus T cells. Unlike our TRM bouncers, these cells sit paratumorally.
Paratumor. So they're physically stuck on the outside borders of the tumor.
Exactly. They never make it inside.
And when the researchers looked at who these outside T cells were hanging out with, it was a really bad crowd.
Oh, it was a highly immunosuppressive neighborhood. They were clustered right next to TREGs, which are regulatory T cells that exist to tell the immune system to stand down and prevent autoimmunity.
So they're getting bad orders.
Yep. And they were also clustered with MRC1 plus TAMs.
Okay, TAMs are tumor-associated macrophages. But I thought macrophages were essentially just the immune system's garbage collectors. You know, they roam around eating dead cells and debris. Why is the tumor interacting with them at all?
Under normal circumstances, yes, they are the cleanup crew. But tumors are incredibly manipulative. They secrete chemicals that hijack the macrophages, fundamentally changing their polarization.
So they brainwash the garbage collectors.
Basically, yeah. That MRC1 plus subset is a specific type of hijacked macrophage that actually helps the tumor hide. It repairs the tumor's blood vessels and secretes molecules that paralyze those T cells stuck on the outside.
Let's pull these threads together for a moment just to keep everything straight. We've got 5 tumor ecotypes. In the favorable ones, we find these elite TRM cells. They use the CD103 anchor to permanently embed themselves deep inside the tumor, actively fighting and turning the tumor's suppressive signals into residency permit.
Perfect summary.
Meanwhile, the other T cells are stuck on the outside, surrounded by hijacked garbage collectors and regulatory cells that just shut them down.
It's a tale of two neighborhoods, really.
So if you are listening right now and picturing this battle in your mind, think about your own immune system. Would you rather have a massive, confused army waiting outside the city walls, constantly being lied to by hijacked double agents? Or would you rather have a tiny elite squad of bouncers permanently stationed right inside the enemy's headquarters?
I think we all choose the embedded bouncers, without a doubt. It's a highly effective strategy.
Definitely.
But what this study reveals is that the immune system actually has more than one way to win. TRM cells act as elite solo operatives. But sometimes the body decides it needs to bypass the solo operative approach entirely.
And do what instead?
Build an entire coordinated military base right on the front lines.
Yes. And this brings us to the second major hero of our story, found mostly in ecotype 5, tertiary lymphoid structures, or TLS. Unpack that for us.
A tertiary lymphoid structure is a highly complex, organized cluster of B cells and T cells that spatially accumulate around blood vessels.
Okay, but what does it actually do?
Well, normally, your immune cells train, coordinate, and share intelligence in your actual lymph nodes. Think of the glands in your neck or under your arms. But a TLS is essentially the immune system biologically mimicking the structure and function of a lymph node directly inside the brain tumor.
That's wild. It is literally a rebel base camp. Deep behind enemy lines, the immune system is constructing a fully functioning forward operating base to train and deploy troops locally. But how do they even build something that complex inside the brain?
It requires highly specialized cellular commanders. When the researchers mapped these TLS formations, they found mature DCLMP plus dendritic cells.
Dendritic cells, those are the intelligence officers of the immune system, right?
Exactly. Their entire job is phagocytosis. They capture pieces of the dead tumor, chop them up, display those cancer proteins on their surface, and present them to the T cells, so the T cells know exactly what the target looks like.
Here is a mugshot of the bad guy. Go get him.
Exactly that. But they aren't working alone to build this base. The researchers found a completely newly discovered subset of macrophages involved too.
Oh, right. We just talked about how macrophages can be hijacked to protect the tumor, but this new subset called CXCL13 plus TAMs does the exact opposite, right?
They do. They're the good guys here. They secrete a specific chemical attractant called CXCL13.
And what does that chemical do? Like, why is it so important?
It acts like a powerful beacon. It actively pulls B cells out of the local blood vessels and draws them into the brain tissue to help construct and fortify this rebel base. It's a localized recruitment signal.
The clinical revelation here is just staggering. When the researchers looked at the patient survival data, the presence of these tertiary lymphoid structures was incredibly predictive of prolonged overall survival. Patients whose bodies managed to build these rebel bases fought the cancer off significantly longer.
They really did. It's a massive survival advantage.
But there is a massive twist in the data here, too. Tumors with high amounts of those elite TRM bouncer cells and tumors with these TLS rebel bases, they almost never overlap in the same patient.
That's right. They represent two completely distinct, successful evolutionary strategies the body uses to fight breast cancer brain metastasis.
So it's an either-or situation.
Pretty much. You either get the intratumoral embedded elite squad or you get the perivascular organized military base. Rarely both.
Okay, so to synthesize this second layer for everyone listening, TLS are makeshift lymph nodes built inside the brain tumor. They rely on dendritic cells for targeting intelligence, and specialized macrophages pumping out CXCL13 to recruit B cells from the blood. And having them strongly predicts you will survive longer.
Spot on.
Which gives us something really profound to chew on. You might be wondering, why does a patient's immune system choose one strategy over the other? Are some tumors just genetically better at blocking the rebel base from being built, leaving those local bouncers to fight alone?
It's one of the most important questions in oncology right now, and researchers are actively trying to answer it.
I bet.
But in the meantime, if we know the body has these two distinct strategies, the immediate question is, what can a doctor actually do with this information today?
Right. Moving from theory to the clinic, can we actively assist these heroes medically? How do we turn this biology into a treatment?
Well, the researchers tested this using something called an organoid killing assay, paired with immune checkpoint blockade, or ICB.
Okay, break that down for us. What's an organoid killing assay?
To see if they could empower these immune cells, they took live breast cancer brain metastasis tissues straight from the operating room. But instead of just growing them flat in a petri dish, which destroys all those important spatial relationships we talked about.
Right, the Google Maps coordinates.
Exactly.
Yeah.
So instead of flattening them, they grew them as organoids. These are 3D mini-tumors that preserve the actual architecture and extracellular matrix of the patient's cancer.
That's amazing.
Then, they reintroduced the patient's own T cells into the dish.
And to understand the experiment, we have to understand a key vulnerability. Those elite TRM bouncers. They are fierce, but they have a built-in weakness. They express receptors on their surface called PD1 and TIGIT. Tell us how those work.
PD1 and TIGIT are immune checkpoints. You can think of them as emergency off switches.
Off switches for the immune system?
Yes.
Yes.
Evolution designed them to stop the immune system from becoming overactive and attacking your own healthy tissue.
Like in autoimmune diseases.
Yeah, exactly. But the tumor is incredibly smart. The cancer cells develop proteins on their surface that physically reach out and touch those PD1 and TIGIT receptors on the T cells. By doing that, they effectively flip the switch and paralyze the bouncers.
So the tumor just reaches out and turns the immune system off. That's terrifying. But the researchers intervene in these organoid models, right?
Yes. They applied a dual ICB treatment, a combination of drugs that specifically bind to and block both the PD1 and TIGIT receptors.
So they covered the switches.
Exactly. By physically covering up those off switches, the tumor could no longer touch them. The cancer cells tried to paralyze the T cells, but the switches were blocked. They essentially cut the brakes on the immune system.
And the result was incredible to read. Massive tumor cell death ex vivo. The TRM cells woke back up, multiplied, and completely decimated the organoid tumors in the lab.
It was a huge success. It proved the mechanism works. But the researchers didn't stop at the petri dish.
Right. They wanted to apply it to real people.
They wanted to know if they could use these granular spatial discoveries to predict which real-world patients would actually respond to therapies. So they took all this incredibly complex spatial and cellular data and translated it into machine learning gene signatures.
Okay, I want to make sure I understand this step clearly. A gene signature is basically a mathematical formula, right? They program an algorithm to look at a patient's bulk RNA data and find the exact pattern of gene expressions that mathematically flags the presence of those TRMs, and another formula that flags the TLS rebel bases.
Precisely. They turned biological neighborhoods into mathematical biomarkers.
That's genius.
It is. And then they tested those signatures against entirely independent groups of patients to see if the math held up. They applied the algorithms to the TAECH Precision Oncology trial for metastatic breast cancer and the massive TCGA primary breast cancer database.
And the results were stunning. The signature successfully predicted patient survival across the board. If your tumor tissue mathematically matched the TRM or TLS gene signature, you lived longer, period.
But here's the most remarkable part of the entire study, in my opinion. These spatial signatures didn't just work for breast cancer brain metastasis.
Wait, really? What else did they work for?
The researchers took the TRM and TLS gene signatures and applied them to databases of patients with completely different types of cancer: melanoma, gastric cancer, and lung cancer.
No way.
Yes. And the signatures successfully predicted whether those patients would respond to immunotherapy. The fundamental biological rules of this immune battleground carry over across completely different cancers.
Wow. So to bring it all together for our final synthesis, growing 3D mini-tumors prove that blocking the PD-1 and TIGIT off switches functionally reawakens the elite TRM cells. And by translating these spatial cartography discoveries into machine learning gene signatures, doctors can now predict survival and therapy response across multiple types of cancer.
It's a huge leap forward.
Think about the implications of this for you listening. Could a simple RNA test for these specific spatial gene signatures one day completely replace the agonizing trial and error approach to prescribing immunotherapy?
That is the ultimate goal, moving medicine from educated guessing to absolute precision. If we connect all of this to the bigger picture, this 2026 study fundamentally changes how we view the human brain.
It really does.
The brain is not an immune desert. Survival in brain metastasis isn't just about random luck or dropping a nuclear bomb of chemotherapy. It is dictated by highly specific, granular, spatial interactions.
It's all about the neighborhood. It's about whether your immune system has the local TRM bouncers embedded deep inside the tumor using CD103 anchors to hold their ground and fight face-to-face.
Right.
Or whether you have the TLS rebel bases using CXCL13 chemical beacons to recruit B cells and coordinate a massive localized attack.
And it highlights that modern immunotherapy works, but it only works if you are empowering the right cells in the exact right spatial neighborhood.
Exactly. So for everyone listening, the next time you read a headline about a miracle new immunotherapy drug, remember this deep dive. It's never just a magic bullet. It's about understanding the specific ecology of the tumor. It's about giving back up to the bouncers or sending vital supplies to the rebel base.
Beautifully put.
Which leaves us with one final provocative thought to mull over. If we now know exactly what the winning strategies look like, if we know that TRM cells and tertiary lymphoid structures are the ultimate biological keys to surviving a brain metastasis, could the future of medicine involve a vaccine?
Well, a preventative approach.
Yeah, a preventative treatment that actively forces your body to build these rebel bases inside the brain, setting up the ultimate defenses before a cancer ever has the chance to spread there.
Now that is a future worth fighting for.
The fortress isn't empty. We just have to help the guards.
Thanks for joining us on the Deep Dive.